recording? Alright.
You ready?
Hold on, let me get this glare off of it. Alright.
Just click play.
Oh, shoot.
Be up on stage and this is gonna happen. Oh, what the hell?
No.
Yeah, his kid's gonna be running it, don't worry. Who's kid? Rich.
What is synthetic biology? Biology is technology. Metabolism is an information process. Life is software, digital code that can be hacked. Metabolism is an information process. Genes are linear sequences of digital executable code. A tree is running a software program on a chemical processor that inputs water, carbon dioxide, energy, and outputs cellulose, or wood. Wood is created from the remixed atoms of air and water. So why can't we hack the system and reprogram the software of life? We can and we do with the aims of producing materials, polymers, energies and fuels, pharmaceuticals, food, water, structures, and architecture. Imagine being able to do all of this, but not with the brute force techniques the chemical industrial complexes do today, using extreme temperatures and pressures, harsh acids, toxic chemicals. With biology, all reactions occur at standard temperatures and pressures, using natural products with no unwanted byproducts, and it all runs on sugar and sunlight. All we have to do is learn to program nature's chemical nanomachines, and the software literally builds the hardware, and the hardware is self-replicating. Engineers want to standardize biology for ease of engineering. Like Lego blocks, you have biobricks, modular parts that connect together into universal supporting chassis such as yeast, bacteria, E. coli, viruses, bacteria. There is now the Registry of Standard Biological Parts, a catalog of parts that build and plug together and snap together, all with open source code for DNA hackers and the biopunk community to experiment with. Much like the Homebrew Computer Club, where kids were experimenting around in the garage, which gave rise to Apple, biopunk today are laying the foundation of the future microsoft that will bring about the second industrial revolution. New biohacker spaces are popping up everywhere. You have iGEM, the International Genetically Engineered Machines competition, where you have the world's best gene hackers meeting up and exchanging ideas, all of which are undergraduate students or younger. You have people like creating you have people creating microbes that sequester arsenic. You have and heavy metals and waters. Hack the live bacteria in yogurt to produce Prozac. Cancer-fighting beer. THC-producing yeast. Landmine-detecting flowers. Yeast cells that are LED arrays. Bacteria that grow cracks back together in buildings. Mitchell Joe Hockham and Rachel Armstrong and others are designing whole buildings and cities that grow and repair their own architecture while adapting to local conditions and cleansing the environment around them. Angela Belcher is using viruses to grow batteries and diatoms to grow glass, and the aims to get microbes to grow entire photovoltaic solar panels. Alchemy paved the road to chemistry, which led to the whole synthetic organic chemical industrial complex. This is what we need for synthetic biology. Biology is now an information science. Using math and formulas to yield predictive powers, we can model and simulate biology and genetics, ultimately being able to design and engineer entire systems before constructing them. Genome compilers, protein folding predictions, mail-order DNA are just some of the things being designed and abstracted for genetic engineering, with drag-and-drop components and features. Places like DIY Bio, GenSpace, BioCurious, Hackteria, and other biohacker spaces are springing up all over, where you don't need a degree in biochemistry to splice genes, just passion, curiosity, adventure, and imagination to be a citizen scientist. Biopunks feel access to information and science shouldn't be limited to the few elite scientists in their ivory tower with expensive lab equipment. So we are developing low-cost equipment with off-the-shelf parts, democratizing science to all. I've repeatedly gone to iGEM, visited GenSpace, met with the people at the forefront of this field, such as Andrew Hessel, Drew Endy, Rob Carlson, even Nobel laureate James Watson. I've seen and done it all, and I'm tired of sitting on the sidelines and being just a fanboy. So I've started up my own lab. I've prototyped an incubator, a centrifuge, gel electrophoresis box. I've bought a set of pipettes on eBay. There's future plans of building a PCR machine from the Open PCR project. I'm passionately curious and don't have time to wait for answers. How would you like to hack the software code of life? How would you like to work with a 13.7 billion-year-old engineering toolkit? I invite you to help me start a lab where we can empower individuals and put the tools in as many hands of people as possible. Where biotechnology is social, global, ubiquitous, and cheap. One where we can design and engineer a world where anything is possible and we are only limited to our imagination. Be a scientist. Change the world.
How's the timing run?
a little bit fast because I couldn't see it. But this would be the last thing right here.
All right.
If I could see it, I would know where
Right,
I
you
was.
mean
Yeah.
You'll be able to see it right there.
Yeah, yeah. I thought I was ahead for a little bit.
Ignite setup.
Can you see that? Zoom in on that. The
We-
quote so you can read it.
We take the slides into there. and then
Yeah.
the screen is in front of the
Dude, that was
They
an
got
atrocious
a
camera
monitor
work.
down low.
Was it? Oh, no.
Just like back and forth.
Oh, you should have just kept it on this. I just wanted to talk over this.
Giant
This
No.
wasn't that exciting.
What? You needed to zoom in on that. All right, let's
I mean,
stop
I got
this.
every frame.
It's
How do you stop recording?
so you click the big stop button.
I
You're
don't-
smart ass, but
I'm not seeing a big stop button. I'm sorry.
Is it the red button?
No, that's camera one. That's input.
Is cut?
Ethan, not being a smart
Is that
ass.
gonna be